Hello, Munchkins. So, this is going to be your lecture. So, before you move on to action potential, you need to review what we talked about last week, which is step one, which is the resting membrane potential. So, these are the things that you are expected to know and should review for the resting membrane potential. This was also went over in your guys' homework assignment from the weekend. So, reminder that the resting membrane potential is when a neuron is at rest and not excited. And we talked about the fact that the outside of the neuron, the outside of the cell, and you guys can see that here, is more positive than the inside because there's so many positively charged sodium ions on the outside. So if you guys look at this picture, there's the key up here. The little circles are sodium ions. The little diamonds are potassium ions. And so we can see that there's a lot of sodium on the outside, a lot of potassium on the inside, but there's even more sodium on the outside. So that causes the outside to be positive compared to the inside. And so the resting membrane potential is said to be at negative 70 millivolts because it's more negative on the inside because there's less positive stuff. So you need to know where more sodium is and the direction that it's going to diffuse in, where more potassium is, and which direction it's going to diffuse. So remember, sodium is going to diffuse in if it can, because it's in higher concentration outside. Potassium will diffuse out if it can, if the gates are open. And then we talked about the sodium potassium pumps. And so that's this little orange guy over here. And so remember, sodium potassium pumps use active transport to move the sodium and potassium where we want them to be. So it moves the sodium back out to the outside of the neuron and the potassium back into the inside of the neuron, so it can maintain that negative 70 millivolt charge. So we will talk about this in a little bit more detail and go over questions from your guys' homework when I get back to class on Tuesday and Wednesday. But those are the key things that I want you guys to know from what we talked about last week. So now we need to talk about step 2, which is called an action potential. And so what we're going to talk about for the next couple of days is nerve impulse transmission. And so step 2 is called an action potential. And this occurs after the resting membrane potential in a neuron is established. So it has to be at that negative 70 millivolts. And then an electrical impulse is generated in the dendrites and sent along the axon to the axon terminals. And then next class, we'll talk about what happens once it reaches the axon terminals. But we're just going to focus on this one part because there is a lot of steps with it, and it tends to be confusing for kids. So all of the resources that are in your Schoology folder, please make sure you guys are using them because this can be one of the harder things for students to grasp because it's not memorization. We have to look at the whole picture and how this whole process works because it's happening inside of your body as we speak, as you guys are taking notes. So it's not just memorization. It's not just separate things. We need to understand the whole picture. So an action potential, you guys will see me shorten it, AP, is also referred to as an impulse and is when a neuron sends information down an axon away from the cell body, because remember, neurons always travel in one direction. It's like a one-way street. And an action potential is an explosion of electrical activity caused by a depolarizing current. And so we're going to talk about what that depolarizing current means and how it works in a neuron so that it can communicate with other neurons or other effector cells. So you might be wondering, how does an action potential start? And so for an action potential to start, there has to be some type of stimulus. So maybe it might be light, sound, touch. There might be something that our nerves perceive. And when they perceive that, it changes the membrane permeability of the dendrites that detect that stimulus. And so what happens is when there's that stimulus, it then opens the sodium channels in the membrane of the dendrites to allow sodium to come in. And because there's so much on the outside compared to the inside of the neuron, that sodium is going to rapidly diffuse into the cell through the sodium gates. And then this causes, probably can't read that. It causes the resting membrane potential to move towards 0 millivolts because all of that sodium is rushing in. Because remember, ions have a charge. It lost an electron, and so it actually has a charge. And so if all of that positive is rushing in to the neuron that was originally at negative 70, it's going to cause the inside of the neuron to increase towards 0 because the sodium has a positive charge. And so it causes the inside of the membrane, the inside of the neuron to move more towards 0, since all of those sodiums carry a charge with it. And remember, guys, if I'm ever going too fast and you need to write things down, just make sure you pause. You can rewind, you can watch it. That's why I told you guys to bring your earbuds so you didn't have to worry about trying to watch this all as one class. So then what happens is if enough sodium enters through the gates, it has to reach a threshold potential of negative 55 millivolts. So enough has to go in to get it above negative 55. And so that's going to be one of those things that you guys are going to have to remember is the threshold potential. And so if the threshold is reached, then depolarization will occur and it'll cause more sodium gates to open, allowing more sodium into the cell and reversing the polarity of the cell where the stimulus was applied. And so if the stimulus is of adequate intensity, the depolarization wave will travel rapidly down the rest of the membrane to the axon. So what this means is that if the stimulus is enough intensity to open enough sodium gates, so that enough sodium comes in to get the inside of the neuron above negative 55 millivolts, then it causes a domino effect all the way down the axon to the axon terminals. Because what happens is once one sodium gate's open and they have that adequate threshold, it signals the next sodium gates to open. And so more sodium rushes in, which signal the next sodium gates to open, which causes more sodium to go in. And so that's why when we watched that video on Thursday, Friday last week, you guys saw the electric charge travel down the neuron. And that was because of this. It's that domino effect. That's what we were seeing in that video. And if not, then the action potential will not fire. And this is a key part of the nervous system because we don't want our neurons firing all the time. That would not feel good and it would not be efficient. And so that's why we have that threshold potential so that we're not responding to every single stimuli in our lives. And that our neurons are not always firing all the time. And so here's a nice picture to show you guys. Here's your resting membrane potential that we've talked about before with lots of sodium on the outside, a little bit of potassium. And then on the inside, there's less positive stuff, but there's more potassium. And so during the depolarization, you guys can see here this blue sodium channel protein open. And so since there's so much sodium on the outside, it rushes in, causing the inside of the neuron to get more positive. And so you guys can see it can get all the way up from negative 70 millivolts inside the neuron to plus 30 millivolts inside the neuron. And so you guys can see one of the other things that happens during depolarization is the potassium channels close so that no positive ions can move out. And then the sodium potassium pumps are also closed so that only sodium is rushing in to cause that generation of the electrical current down the axon. This is going to be one of the figures that you guys are expected to know for your quizzes. And so what we've just talked about, clicked too soon. What we've talked about so far is step 1. So it shows you here that the stimulus is applied. And so sodium gates open up due to the stimulus. And so that causes sodium to rush in. And so if it gets above that gate threshold here at negative 55 millivolts, then even more sodium gates will open to cause depolarization. And again, this will happen all the way down the axon as a domino effect, so one after the other. And then eventually, after the action potential passes, which we're going to talk about next, the sodium gates closed. And so we're going to talk about the second half of this graph now. So the next part is called, I keep having to move this, repolarization. And so this is after the sodium gates have opened and all the sodium is rushed in. We don't want that neuron to keep firing, we need to reestablish the resting membrane potential. And so the first step to reestablishing the resting membrane potential is repolarization. And I'm sorry, my dog is playing with her Chewy toy. All right. So during repolarization, the sodium gates, we've talked about that. So during repolarization, after the sodium gates have opened, the sodium gates open slower, but when they do, when the potassium gates open, they open slower. So they open after the sodium gates. And what this does, it allows the potassium to rush out of the cell, which causes the inside of the neuron to go back to zero. So it reverses the polarity and it causes it to go back negative. So it's reestablishing that negative 70 millivolt inside of our neuron. And it's called repolarization, and it occurs immediately after depolarization. So you get a domino effect of depolarization, all of the sodium gates opening one right after the other, because they don't open all at once. It is a domino effect. It travels down like a wave. And then after that passes, that's when we see these potassium channels open, and the potassium will diffuse out of the neuron. And since potassium is also positive, it helps to move that positive charge back out to re-establish that negative 70 millivolt charge that we want for our resting membrane potential. This repolarization happens all the way down the cell as a wave, just like we saw with the depolarization. And the rapid change of the polarity of the cell from negative to positive back to negative is the whole action potential. That's the whole electrical component of the electrochemical response that neurons employ to be able to communicate. Like I said, this occurs down the cell like a domino effect. And so now we have this period called the refractory period. And so it's after the action potential has passed, and it's there to reestablish the resting membrane potential. So it goes extra negative. So more potassium leave. And so it goes even lower than the negative 70 millivolts. And so that's just to ensure that another action potential can't be set until the entire neuron is back at its resting membrane potential. And the sodium potassium pumps are used to reestablish the sodium potassium balance so that we get to the negative 70 millivolts inside of the neuron and it gets reestablished. So that's what this looks like on the graph. So sodium gates close. And so you guys can see we're all the way up here at plus 30 millivolts. And so what happens then is that signals the potassium gates to open. And so because there's more potassium on the inside of the neuron than the outside, it is going to diffuse from the inside to the outside. And because it's a positive charge, it's going to carry that positive charge with it. And so you guys can see here that so the red line is showing you the charge inside the neuron. And so we can see here as those potassium gates open, it drops down. So here's your repolarization. And then this extra dip here is called hyperpolarization, where it drops even lower. And that's just to ensure that no other action potentials can be sent down the neuron until we reach that resting membrane potential once again. And so the sodium potassium pumps help to regulate the concentration of sodium and potassium during repolarization as well as during the resting membrane potential. So again, you guys will have to be able to describe what is happening in this graph in terms of resting membrane potential, the stimulus, depolarization, repolarization, and then what is happening to those sodium potassium channels or the gates, as well as the sodium potassium pump during these times. And so here's your overview of the process as well through the pictures that we've been looking at. And so here's the resting membrane potential. So this is the inside of the neuron, that's the outside. So remember, at the resting membrane potential, we're at negative 70 millivolts. So here's the negative that's showing you the negative charge on the inside. and positive charge on the outside. Some stimulus happens to open those sodium gates. And since they are positively charged, they take that charge with them to cause the inside of the neuron to become positive. That's the depolarization, which generates an action potential. Once that has passed, right behind it, the potassium gates will open. And because there's more potassium on the inside of the neuron than the outside, they diffuse out, carrying that positive charge with them to reestablish that resting membrane potential. And then the sodium potassium pumps keep that negative 70 millivolt inside the neuron by moving any sodium that leak in back out and any potassium that leak in back inside. And so again, there's the graph for you guys. And I like this graph too because it shows you. So when the stimulus is applied, so this graph is great because it shows those failed initiations that we talked about. So if a stimulus is not of adequate intensity and it doesn't open enough, it doesn't cause enough sodium gates to open. So you can see here that the depolarization doesn't happen. So an action potential cannot be generated because the stimulus was not of adequate intensity. And remember, that's important so that our neurons are not responding to every single stimuli that we encounter. There's a lot going on around us, and we don't need to respond to every single thing. And there's the graph again for you guys. So you can use any of these to help you study. They all show the same thing. The last thing that I need to talk to you guys about is the difference between myelinated and unmyelinated fibers. I've talked about it a little bit before when we learned about what the myelin sheath does. And so when we look at the impulse conduction, so when an action potential travels down a neuron, if the neuron is unmyelinated, meaning it doesn't have any of that fatty myelin sheath covering on it, it has to conduct the impulse over the entire membrane surface. So unmyelinated fibers tend to have the action potential take longer to travel down compared to the myelinated fibers because it conducts impulses from node of Ranvier to node of Ranvier. And this phenomenon of the action potential jumping from one node of Ranvier to the next is called the saltatory conduction. And so saltatory conduction is many times faster than the conduction, so how fast the action potential travels down the unmyelinated neurons. And so we'll see later in the unit where myelinated and unmyelinated nerves are in our body and why it's important that we have some unmyelinated ones and why we also need myelinated fibers that can conduct those action potentials faster. And so this is just a diagram to show you guys how that saltatory conduction works. So because the insulation, the electrical charge can't travel through the insulation. You guys can see there's no gates there. And so what happens is the depolarization skips along the action from one node to the next. And it just allows it to go that much faster because it's not having to open gates all the way down. It just opens the gates in between each of the myelins in the node of Ranvier's. So there is a video that you guys can watch. You can't click on it from here, but in the daily agenda slides in Schoology, you guys can watch this video. There's a little bit more info than you need to know. So focus on what we've covered, but it has a great review. It'll allow you guys to visualize, and then just make sure you visit the resources in this week's folder too. And I'll add to the resources as we go through this week. But if you guys need anything, if you're confused, want help, send me an email. But once you feel comfortable with the notes, then you guys can start working on the rest of the pages in your notebook that are assigned to you on Schoology. And then that way, they're just going to help you guys practice this and help you guys visualize everything that we just talked about in these notes. So again, email me if you guys need anything, and I hope you have a wonderful day, and I will see you on Tuesday or Wednesday.